Neural correlate of visual familiarity in macaque area V2 by Haley Dunster. According to this study, neurons in the ITC, or macaque inferotemporal cortex, are more likely to fire when a subject is shown new and complex images than with something they are familiar with. This allows the brain to make sense of new images for future identification as familiar. Macaque monkeys are used in this experiment and many experiments because of their similarities to human brains and their data reliability. In order to test if the neurons in a lower visual area, like the V2, show familiarity suppression, which is a tendency for neurons to respond weaker to familiar images than novel images, semi-chronic microelectrode arrays were used to record activity in the ITC in monkeys while they were shown a set of complex images. From this, it was determined that V2 neurons do exhibit familiarity suppression. This suppression continues developing over multiple days with an exponential growth rate constant of about 100 exposures. In comparison to the ICT, familiarity suppression latency following the images is shorter in the V2 than in the latter. Familiarity suppression in the ITC is characterized by the neuron's decrease in response strength as an image is rendered familiar by long-term experience. In previous studies, familiar suppression has been tested using hundreds of thousands of images over the course of a few weeks. Familiar suppression occurs whether or not image exposure is appealing to active discrimination or passive viewing. Familiarity suppression provides advantages both behaviorally and perceptually through reducing the responses, bettering the signal detection for familiarity, better discrimination of a familiar image, and reduced salience for familiar images rather than new. The ITC was thought to be responsible for familiarity suppression because the neurons in this area are known to have wide receptive fields and appear to be selective for complex images. This assumption could be flawed, as other areas, like the V1 and V2, could also have neurons that code for complex imaging. This experiment's goal is to understand how the activity of the neurons in the ITC helped with familiarity suppression. But another trial recorded activity in the V2 in order to determine how responsible that area was as well. 2 adult rhesus macaques were used for this experiment, making sure that the experimental procedure was approved by the Carnegie Mellon University Institutional Animal Care and Use Committee. 50 images of natural and man-made objects were presented to these animals, 57 centimeters away on a CRT monitor. Each trial showed the subject a picture with a fixation point for 500 to 800 milliseconds. After an additional 200 milliseconds, the fixation point was shifted, and if a saccade occurred, was recorded. Semi-chronic microelectrode recording was used to analyze multiple neurons and average out noise from other areas. 6 experiments were performed to guarantee results. 2 for familiarization and comparison, while the others were manipulated to test how dependent the brain was on their image properties. Receptive field mapping was used to find which neurons were firing and where that line stopped. Images were decoded and the data was statistically analyzed using MATLAB. This table explains what was used in each trial, from which monkey to the number of sessions and whether or not the data was significant. Each row represents another variable that was tested. Row 11 specifically shows what was discussed in the materials and methods section, that the aperture was varied in the last 4 sessions. Using the electrodes, the V2 neurons were recorded, producing 1 to 2 action potentials when stimulated. The data was translated to a histogram that showed the difference between the familiar images and the new ones. It was seen that during the first few experiments, the V2 appeared responsible for familiarity suppression. A familiarity index was developed to help compare each session, with N and F reflecting the mean spike rate of recorded neurons caused by familiar or novel images, hence the N and the F. The index for all experiments was found to be positive after all 50 images were shown. Later sessions were then created to test repeating images. The familiarity index for these later sessions was stronger, which meant that the repeated images were no longer being interpreted as novel images. Aperture was also tested in later sessions, with the results yielding a decrease in familiarity suppression. This could be due to the fact that some of the neurons used were not being stimulated as their perspective field was outside of the focused point. The ITC comes back into play in terms of familiarization and selection. This idea was analyzed by ranking each neuron from best to worst using the following equation. Figure 1 shows the receptive fields of the neurons in the V2. B shows the receptive field relative to the size of novel and familiar images. Figure 2 shows a plot of the different experiments and for how long the monkey saw each image. The line of best fit in Figure 2a is a function of whether the monkey had seen the Figure 3 focuses on the latency of familiar suppression. In 3a, the mean firing rate was graphed showing familiarity suppression was present. 3b showed the difference in firing rate between the trials with familiar images versus the new ones. The final figure in the results section shows how familiarity suppression has some dependency on what is in the periphery of the perceived. The main takeaway from this study is that neurons of the macaque area V2 exhibit familiarity suppression, which was previously thought to only be found in the ITC and its higher order projections. The difference between these two is that in the ITC, familiarity suppression arises from neuron fatigue, whereas the V2 has neurons that are selective for features and will start to become familiar as they see them more often. Though the study wasn't able to determine the real mechanism of familiarity suppression in the V2, there are some speculations. The first option is top-down feedback from the ITC back to the V2. Table 2 shows how the current study's results differed from those in past studies. Population histograms from multiple studies were compiled within this table. Figure 5 in the discussion section shows how the latency of familiarity suppression is shorter in the V2 than in the ITC. A represents a comparison of responses. Based off the information found in the study, it is interesting for us to look further into research to determine just how neurons selected for local features detect a global image as familiar. We believe that it's probable that neurons in the V2 are specific as a group instead of as individuals. In other words, groups of neurons will fire when they see something familiar instead of just a singular neuron being encoded. It is also true that we are not entirely sure of the rate at which familiarity suppression develops in the ITC, though we are aware that it's quite rapid in the V2.